You know, in the world of genomics, there's this quiet revolution going on. And today, we're going to dive into the epic showdown right at the heart of it all, a battle between two giants of DNA sequencing, Pacific Biosciences and Oxford Nanopore.
So picture this. How would you read a book, a really complex one, if the only copy you had was put through a paper shredder? You got millions of little snippets, just a few words each. How on earth are you going to piece that story back together, especially the really long, repetitive parts?
Well, for years, that was pretty much the exact challenge facing anyone trying to read a genome. The older, short-read technologies, they just created this massive, super complicated jigsaw puzzle. Yeah, they gave us tons of data, but trying to assemble the whole picture, especially the tricky parts, was a nearly impossible task.
But then, everything changed. A whole new generation of technology showed up that could read, really read, these huge, long stretches of DNA all in one go. It's like going from confetti to reading whole chapters at a time. Suddenly, the story of the genome became a whole lot clearer.
And that brings us right to the two major players, the real game changers in this long-read sequencing world. They both get to the same finish line, but what's so cool is they take completely different roads to get there. All right, to make this stick, let's think of them as characters. On one hand, you've got PacBio. Let's call them the precision artist, just totally obsessed with getting every single detail, every letter, absolutely perfect. And then, in the other corner, you've got Oxford Nanopore, or ONT. They're the endurance explorer. They're all about covering massive distances, trekking across these huge, unknown parts of the genome in one shot. Okay, so let's get into the nitty-gritty, because how this tech actually works is, well, it's just plain cool. You've got these two totally different approaches under the hood. So first up, let's talk about PacBio and their smart sequencing. The magic ingredient here is an enzyme called DNA polymerase. You know, it's the little natural machine inside our cells that's constantly copying DNA. What PacBio did, and this is brilliant, is they basically set up a camera and filmed that enzyme doing its job, one molecule at a time, in real time. So here's how it goes down. They take one of these polymerase enzymes and stick it at the bottom of this incredibly tiny well. Then, as it starts building a new DNA strand, it grabs these DNA bases that are floating around, and each one is tagged with a different colored light. A super sensitive camera is just sitting there, recording every single flash of light. But here's the real genius move for getting that insane accuracy. The DNA they're reading is actually a circle, so the enzyme just keeps going around and around, reading the same sequence over and over. By doing that, it builds this super strong consensus, and that's what they call a HiFi read. Super, super accurate. All right, now let's flip the script completely and look at Oxford Nanopore. They don't mess with enzymes or cameras or light, nope. They use this tiny little biological hole, a nanopore, and electricity. Yeah, electricity to read DNA directly. Just think of it like this. You're feeding a thread through the eye of a needle. A little motor protein grabs a single strand of DNA and just pulls it, one base at a time, through this nanopore. Now, while this is happening, there's an ionic current flowing across that pore. And as each DNA base, A, T, C, or G, squeezes through, it disrupts that current in its own unique way. So you get the stream of electrical signals. They call them squiggles, which is awesome. And then some really powerful AI decodes those squiggles back into a DNA sequence, all happening in real time.
Okay, so we have these two wildly different methods. What does that actually mean for performance? This is where it gets really interesting. Let's put them head-to-head and look at the real-world trade-offs between precision and, well, pure endurance.
So this table really breaks it all down for us. Okay, let's start with accuracy. PacBio's HiFi reads they're just beautiful. Over 99.9% accurate right out of the gate. Now, ONT can get there too, and even higher, but it does it by rereading the same spot over and over to build a consensus. But then, look at read length. This is a big one. PacBio gives you these really solid reads, like 10 to 20,000 bases long. That's great. But ONT? Oh man, it's in a whole different universe. We're talking single reads that can be mega bases long, millions of bases. And finally, raw data output. The amount of data you get from a single run. ONT's big machine, the Prometheon, is an absolute beast. It just churns out way more data.
So for PacBio, this number is everything. That greater than 99.9% accuracy. That's not an average, that's on a single read. That makes it the go-to tool when every single letter has to be right, like when you're hunting for tiny genetic variations that might cause a disease.
And on the flip side, this number, this is ONT's superpower. A single read that's over 1 million bases long. I mean, that's our Endurance Explorer in action, right? It's just striding across these gigantic, messy, repetitive parts of the genome that other technologies just can't even touch.
All right, so all this tech is amazing, but it leads to the big question for any scientist. Which one do you pick? And as you've probably guessed, the answer is, it totally depends on what your goal is. It really just comes down to this one simple question for your project. What's more important to you? Is it getting the most perfect, error-free sequence imaginable? Or is it building the longest, most complete map of the entire genomic landscape? If your answer is, I need accuracy, I need perfection, then PacBio is your champion, hands down. You'd use it for things like finding those really tricky structural changes in a cancer genome, or getting a crystal clear picture of all the different RNA messages a gene is sending out. Anything where precision is absolutely paramount. But if what you need is insane length, or maybe you need your data right now, or you need to do your sequencing out in the middle of a field, well then ONT is your go-to. I mean, you can literally track a viral outbreak as it's happening. You can take their little mini-on sequencer anywhere in the world, and it's the only way you're going to solve those super complex, super repetitive parts of a genome. Plus that whole direct RNA sequencing thing, that's a huge, unique advantage. But you know what? Here's the final twist in the story. For the biggest, toughest challenges in genomics, the real winning move isn't picking one or the other. It's about getting them to work together. That's right. It's called a hybrid strategy, and it is literally the best of both worlds. You get the Endurance Explorer to map out the territory, and then you bring in the precision artist to paint in all the perfect details. It's an unstoppable team. So here's how that works. Step 1, you use ONT's crazy long reads to build a rough but complete scaffold of the genome. You get the whole structure down. Step 2, you come in with PacBio's super accurate HiFi reads and basically polish that scaffold, correcting any tiny mistakes and filling in the sequence with absolute confidence. And the ultimate prize? A telomere-to-telomere assembly. We're talking about a completely finished, gap-free blueprint of an entire chromosome from tip to tip. That's like the holy grail of genomics. So when you step back and look at it, we're at this absolutely incredible moment in science. Because of these two amazing technologies, both competing and helping each other, the question isn't if we can read the full blueprint of life anymore. We can. The new, and I think much more exciting question, is how do we choose to read it? What story are we trying to tell?